It's like an itch. Like certain problems that I wish to solve just bother me. They come out of nowhere, and for some reason I want to find the answer to them, and I can't forget them until I solve them. And that is really what, for me, drives research, right? It's literally, you feel like you need to scratch, and until you scratch it, you can't let go.
Welcome to People Doing Physics, the podcast that explores the personal side of physics at the Cavendish Laboratory, the University of Cambridge.
Hello, I'm Charlie Walker, an astronomer at Cavendish Astrophysics.
And I'm Vanessa Bismuth, a communications manager at the Cavendish.
Today in People Doing Physics, we're delighted to welcome Professor Teuta Pilizota, a physicist who works at the intersection of biology, chemistry and physics. Having set up her first lab during her PhD, Teuta is no stranger to the challenges of moving labs, and she recently brought her innovative biological physics work to the Cavendish Laboratory. Teuta brings a unique perspective to science. She's someone who finds the complexities of maths more straightforward than those of human nature, and who will choose excitement over logic when selecting scientific questions to research. Her curiosity drives her to scratch the surface of living matter and to seek out new physics that is not yet understood. Together today, we talk about resilience, patience, perseverance, and what to look for in the basement of a physics lab. Stay with us. So welcome, Teuta. Thank you for being with us today.
Thank you for having me.
So as always, let's start at the beginning. Your childhood and early education took place in Zagreb. Could you tell us a little bit about what you liked at school and what you didn't like?
So I liked at school, I liked primarily math. That is one thing that I clearly remember liking. I did well in other subjects, but it was just quite straightforward to memorize, so I can't say that I particularly liked them. I like drawing as well, but I like math more than I like drawing. I found math very calming.
And do you think that your interests were influenced by anybody, perhaps your parents? Can you tell us about what they did?
So my mom is an architect and my dad is a mathematician who later on moved into informatics. I think I was influenced by my mom in that I thought quite rationally as a child, if I'm good at math, I'm good at drawing, I should be an architect. And that's pretty much where I stopped thinking about what should I do with myself in the future. It was kind of like solve problem. So I didn't sort of pay attention to whether I like something and where I should take it into a career. I would like something and do something, but didn't really think about it as a career choice because that I already ticked off.
But you didn't mention as well that you didn't really like biology. Could you expand on that?
I didn't particularly like biology at all.
Why not?
Although I love the people who taught me biology, the two professors, they were fantastic. Well, first of all, I thought that I had all the diseases that we ever studied about. And second of all, it was just a lot of memorizing. So there was nothing intriguing about it.
But so you were like setting off to become an architect, but clearly if we're talking today together that has changed somewhere. Could you tell us more about that change? When it happened? What happened?
Yes, certainly. So it changed 2 weeks before the entrance exam to the architecture department, where I was preparing for it. You had to prepare the math exam and a drawing exam, and I think there was something else like history of art. And I remember I was drawing a chair in my room, and I realized that I don't want to be drawing for the rest of my life. And this was then like, okay, what do I... And it was the first time where I went back to think about what I actually like, now having discovered that I don't want to be an architect. And I realized that, well, I really liked math, and I actually liked physics. I was part of a lot of, having grown up in Eastern Europe, a lot of these competitions that are organized on a city, school, and then later Olympics level. So if you're good at it, you get sent, even if you don't think of it as a career path. So as part of that, I did one that was experimental. I was looking at diffraction of gold beads with a Heine laser, which is something that I still do in the lab. And I really enjoy that. So I decided that this is probably what I should be doing. However, I haven't prepared for the entrance exam. So I went to the Department of Physics and I said, what do I need to do It's like only a couple of weeks probably. How does the entrance exam looks like? And they looked at my grades and they looked at all these physics and math competitions that I've done. And they said, you do not need to do an entrance exam. We'll just take you. And this is how I ended up in the physics department.
And so you made that decision in the course of 2 weeks, and
It was
you
in
did
the
the
course of 2
entire
weeks.
swap.
Yeah, it was a total swap. But it didn't feel as drastic as that. And with the first 2 years of the university, I absolutely loved it. I thought this is the right decision. The fact that I'm just surrounded, it was still mostly math at the first couple of years. And then with a sprinkle of physics and everybody talking just about that, I thought like, okay, this is great.
So you started a 4-year undergraduate degree in physics in Zagreb, and then you did a diploma thesis. At the start of your degree, did you know which topic you were interested in and which you weren't? What you were originally seeing yourself doing, if anything?
It kind of came slowly. I don't think originally I was thinking about what I might be doing later. I think I just enjoyed the first 2 years. I didn't know that astrophysics was not something that I was that interested in. And I knew that because quite a few others in the class were. And I found interesting that when we were talking about it, I just always thought it's overwhelming, it's too far. I can't even go there. Right? So it was kind of knew that it was a bit like it felt overwhelming. So I knew that this is not going to be the direction that I'm going to go into. And later on in like 3rd and 4th year, I heard about some of the like molecular motors and how people were starting to understand biological system in a similar way that we do non-living matter. And I found that very intriguing because, okay, this humans and living stuff that I found so confusing, maybe actually it can be made a bit less confusing and more clear in some kind of mathematical form. And that I found very intriguing. So then I started thinking about what I could be doing.
But so did you go straight into the biological physics, and you took some detours, didn't you?
So I didn't really. I still majored in solid state theoretical physics. And I took courses that are related to that. It was also a little bit linked to what was on offer and who was teaching the courses. I took that into account, and I did think, well, solid state physics is the right scale that I want to be. But it took a little bit of time to kind of discover what kind of, what is this biological physics or biophysics, what it really is, because there's quite a lot of work, for example, in functional imaging of the brain. And then I thought, okay, no, brain is too complex, not yet, right? So I really, I was fortunate to take part in Les Houches summer school when I was a 3rd year undergraduate. And I was babysat by some of the people everyone who was doing something exciting in that field was there. I knew nothing about them though. So I was babysat by some of the I'm not sure if they remember, like Nienke Dekker, who continue to do beautiful work in that area, and they were like at a PhD level, a postdoc level. So I really got exposed to what was happening on this level of molecular motors and biophysics of molecular motors, and I thought that's very, very exciting. And I met a couple of people that I thought I could do my PhD with. One of them was actually Tom Duke, who was at University of Cambridge, and a couple of people, Joe Howard, who was then in Dresden, in Max Planck Institute. So it was, I think it was the moment that I realized, well, a couple of weeks, that I realized that this is the direction that I would like to go into. But I was originally thinking that it should be theoretical still. Then I got convinced that given the amount of data in the field at the time, it should probably be experimental. And that also influenced my decision.
Because we were talking about what sort of time, or when was that?
This was the end of third year undergrad. So when was this? Like early 2000, and probably 2000 even.
And so there was not enough data in.
There was not enough data yet. Yeah, it was just kind of the field was just emerging. I would say.
And how did you come to be at the summer school in the first place?
It was recommended to me by one of the professors in biophysics. Selma Supek was there, holding this course, and she recommended that I go, and she convinced me that when I'm undergrad that I should probably take a chance because it doesn't happen as frequently in this topic. And I also think there was this UN aspect, so the fact that I was coming from Croatia and filling the quotas. I think that helped as well, like, to be quite blunt. So,
well, any opportunity is a good opportunity.
Indeed, I do not mind at all.
And you mentioned that the summer school spoke to you in a way that biology at school clearly did not. So what was it exactly that really seized your interest? And set
it was
your
really very
ambitions?
simple, that you could put equations
Yeah.
to the way these molecular motors walk and produce torque and force and all of that. It was this quantification of the living system that I found very, very exciting, because I find it that when you put equations to something, it's very, very orienting. You can kind of like, almost as if you drew a map to something, so you can, turn it whichever way around, and I still feel I know where I am and I understand it. And I don't feel like that if equations are not there. I kind of feel like you could, spin me, and then if you stop me at the wrong moment, I will not know where I am. If there are some equations that describe a system, this doesn't happen to me. I can very, very quickly orient myself, and I feel really, very comfortable with this. And it's almost like a level of calmness when that happens. It's like something when you don't understand, it's almost like an itch you need to scratch. And when you do understand and you formulate it in a mathematical form, it's a sense of release.
And so was that a discovery that you could actually talk about biology in
that's,
equations?
That's precisely what the discovery was. Okay, this could work. So yeah, that and the fact that we could do more and more of that, and some of this, confusing world of living beings could become a bit more comprehensible. Right? That was what I found intriguing and exciting.
So this might also explain the tendency towards theory rather than experimentalism until you were given this extra piece of advice.
Probably, but also we have to take into account that I was doing a degree in Croatia, where the science is not that well funded, which means that those who are doing the science and doing it well will be tending to do theoretical science simply for practical reasons. So it's probably a combination of the two. Mm. Yeah.
And so now you've had a lot of experience with labs and setting them up and moving them around. So how do you feel about that advice now?
Yes, indeed. Having a big lab brings all kinds of challenges that having a computer and a pen and a blackboard doesn't. But I think, yeah, it was the right choice. And actually confirming, seeing the experiments, actually seeing that it does happen is exciting. I would also say that I learned that there is 101 little thing and little idea that you need to implement to get the experiment to work that you don't even appreciate unless you start doing experiments. It kind of seems straightforward, you take a bunch of equipment and you measure. It's not as straightforward as that at all. There's kind of like a lot of little ideas that you're putting in and you're solving it to get the experiments to work. So I enjoy that. Yeah.
And so that was it. You made another decision and to go into biological physics. And you were, after your degree and your diploma, you did look for PhD positions. Tell us about that stage. What were you looking for? Where did you apply and where did you end up?
So yeah, I applied mostly to people who I met at this summer school. So I applied to Tom Duke to Cambridge, and that was a complicated process because I understood nothing about what colleges are, and I had to choose a college. And they all had, the description was lively common room, lots of green grass, and I didn't understand what was a common room and what I was supposed to do with the green grass other than sit on it. So once I understood that process, I thought, okay, I should look at Oxford because it's probably similar, so I'm going to capitalize on the time invested understanding the system. So I did, and I found another person in Oxford who was doing molecular motors. His name was Richard Berry, so I applied to him as well. And then I applied to Max Planck Institute in Dresden, and I think I applied to University of Nottingham as well to work with AFM. And then I got called to interviews a couple of these places, and I selected based on how comfortable I felt in the building and in the surrounding of where the labs were. I kind of felt like some of the interviews, the buildings and the rest of the groups were quite bio. So I didn't feel comfortable. I, in the end, decided to go with Richard Berry in Oxford because I felt like he knows what I know and what I don't know. And a kind of science I feel comfortable with and what I will need to learn. Right?
Yeah. So what do you mean by that? He knows what he working?
So because his background was also in physics, and we were based in physics building, and I always joke about this, that when some of the buildings that I visited during the interviews, they showed us all around the building. There was a lot of bio-related work, like they would have fish in the basement, like rows of fish, and I would feel very disoriented because in basements you keep magnets, right? So I guess that the message was really that I didn't feel like I made a mistake in terms of what I wished to study. I didn't want to go and study biology. I kind of wanted to apply the physics viewpoint onto learning about living matter, and I was very aware that I'm a novice at that, right? That I know very little biology. So I wanted to be in an environment where those that I work with appreciate how little biology I know, but also who know what I do know, right? And that's how I made the decision.
And there were magnets in Oxford.
There were magnets in Oxford basement of Clarendon Lab. There were indeed magnets.
All was well in the world.
All was well, indeed.
Fish were on the roof.
Fish on the roof. No, fish were grilled. Like, well done. Yeah.
You told us earlier that your PhD required a combination of hard work, luck, and talent. Could you explain what was so challenging about your project, and what did you have to do in your first years?
I think what was challenging about my project is probably what is challenging about every PhD project, which is that you're trying to find out something new about the world around you, and you are going. The way you go about it is based on what is essentially an educated bet, a guess. Right? You don't really know whether you're going in the right direction. You have advice from those who have even more experience than you. In my case, more experience than me. I had very little experience in biological physics, if any. So that realization is simply hard, right? Because it does mean that you're exposed to luck by default quite a bit. Right? Versus when you're an undergrad, you get given examples that we know how to solve, and you need to learn them. So if you're hardworking and you're talented, you're going to do well. Here, you can be hardworking and talented, but if you're not lucky as well, it might not go as well. Right? So and handling that element, I think, is probably what turns you into a scientist, but also a grown-up. I found, right, because life has that exact component. In it as well. So it's not just the PhD that requires this amount of luck, other things in life as well. So handling that uncertainty and potential unfairness that you were not lucky and someone else next to you might have been a lot more lucky is part of what the challenge is. I was the second student in Richard Berry's group, which was exciting. So the benefit that I got is that he got to spend a lot of time with me, but it was also a lot of setting up, and it was setting up the optics lab, but it was also setting up the biology lab. I learned how to purify this protein rotary molecular motor that I was working in by going to Japan to a lab, in biochemistry lab in Japan, where I learned the whole purification process, and I had to help set it all up back in Oxford in a building that had magnets in the basement. It wasn't a biochemistry building, so the equipment was not necessarily there, so we had to cross buildings and all that, and that was all challenging. So setting it all up to even get to the stage where you can do the experiments was challenging. At the same time, I learned a lot. Right, I did have an experience of how that all works. Right?
So it was a lot of responsibility setting up a lab for the first time in the first few years of your PhD. How did you find that? And was your supervisor very confident in you?
Yeah, he was probably, I would say that he was too confident in me.
Okay.
And on my end, I would say that I was just naive. I didn't think about it. So a great combination. But I didn't view it as a responsibility in a way, right? I kind of think, okay, these are the things I need to do to get to the place where I wanted to be. So I just kind of plowed through it step by step. Yeah, the project kept going. Right? Yeah. So
and the lab is still there today.
The lab is still there today. Yeah. Well, the microscope that I built got disassembled. I think I might have even gotten an email from Richard to tell me that it's going to be disassembled, which was very kind. But the room is still there and the lab is still there, and there's a different microscope in it now.
And you go back?
I do go back. I went back for I kind of perceived the lab as, where I grew up to be a scientist. So, and I perceived my PhD supervisor as like a science dad. And I went for sabbatical in 2018 for I think 3 months. I tried to do something that didn't fully work as part of my PhD, so I tried to do it again. And this led to, it didn't work either, but but it led to some other interesting things, and it was also good to be back. Yeah.
So then, after the PhD in Oxford, it was time to choose a postdoc. And you mentioned that you were thinking about multiple places. Japan and the US were two of them. So could you tell us a little bit more about that and that decision? What did you choose to do? Where did you choose to go? And why was that?
Yeah. So I wanted to get an experience in US to see how a completely different system works. But at the same time, there was a lab in Japan from Hiroyuki Noji, who just got set up, who was doing these absolutely beautiful experiments on this rotary motor that generates ATP in living organisms. So I was considering going to that lab. I went to visit, and they had everything there, all the microscopes that you can dream to be able to do that work. And I like working hard, so I like the environment, and they grilled fish really well. So yeah, that was it was really a place that I wanted to go to. At the same time, in US, I had a look at a couple of labs, and I identified the lab of Josh Shavitz as the one that I would like to go to. And I think at the time, my original plan was that I first go to Japan and then I come back to US. But at the time, Josh had the funding and he needed someone to come straight away. So then I was like, okay. I think I've seen all the places that I would like to go to in US, and I'm not sure it's going to change in a year or two that much. So I, in the end, decided not to miss the opportunity and to go to US. I was also advised that, from the career perspective, going just to Japan for postdoc might not be as optimal. Although I disagree with that advice nowadays. I think that would have been completely fine. But I had absolutely fantastic time in US as well. I ended up in Princeton, New Jersey.
So did you work very hard and eat lots of grilled fish?
Yeah, well, I danced a lot of salsa. I did eat grilled fish as well. One of my friends from salsa worked in a fish shop.
Yeah.
So there you go. There is a fish team all the way through.
So how was Princeton?
Princeton was a different way of working compared to Oxford. Not drastically different, but there were certain elements that were done differently. I think I'm one of the Europeans who came more with kind of like, sometimes Europeans have a bit of a negative view of US. On average, I would say. Obviously, it varies. And I think I was the one who went relatively neutral, maybe with a bit of a negative viewpoint when it comes to healthcare and some of like social security. But I ended up having a great time and actually having a positive more positive viewpoint after the experience. I was there just for context between 2008 and 2013. Yep. So just to clarify that bit, right? I really like the attitude towards work. I liked, again, that they place work as highly valuable. They work hard, and I like the positive attitude. Again, although you kind of like feel that everything awesome is a bit, sometimes is a bit like, a bit maybe fake, but it's just very energetic. Awesome. So yeah, when I moved back, I did miss the US attitude towards.
And in terms of the research, with the projects, how was it? What did you do? How did it go?
I started working again on in vitro, some of the in vitro work on proteins from one of the bacterium that we have been studying a lot, and it's probably the most studied bacterium, Escherichia coli, E. coli. So the idea was that I will purify this protein, that it's a cytoskeletal protein, and as a cytoskeletal protein, we thought it was going to be structurally stabilizing the cell. So I was going to purify it, measure it in vitro, what kind of mechanical properties it has, and then link that with how it is geometrically positioned in the cell to get almost like a skeleton view of the mechanics of the cell. Right? But in the process, I discovered some things that didn't really fit that picture. And I was working in vitro with these proteins. And I think if at the time there was a group that was working on biochemistry with expert biochemists, they would have probably recognized that I was seeing a signature of this protein being attached to the membrane. Except that there was no one that we talked to at the time. Later on, there was someone actually from Cambridge who discovered that this protein indeed binds to the membrane, and that the way we view what it does changed in accordance to that. So I kind of had all the ingredients to see that, but we didn't see it. So I guess this goes sometimes to say, when someone who is a non-expert looks at a topic, it can come with a very, very fresh view, and sometimes they just don't know what they're looking at. It depends, right? So in this case, I was the one who didn't know what she was looking at. But I did, as I was doing that, and I couldn't make sense of it, I did look at a different regulatory mechanism of this E. coli cell. So during my PhD, I worked also on bacterial flagella motor, which is a motor that is responsible for the swimming of these bacteria. So they have about half a dozen of these motors randomly distributed along the cell body, and then they all bundle up and make a filament, because each motor has this filament attached to it, and this is what propels the cell. And the network that regulates this flagella motor is called chemotactic network, has been really, really well understood. And one could play around genetically with different proteins in this network and really see that this is essentially a system of antennas and feedback loops, really interesting feedback loops from the point of view of engineering to get a very high sensitivity and good response. And then this output that is mechanical in the form of a motor that generates torque. So as I was playing around with this cytoskeletal protein, I noticed that the cells are, swelling and expanding when I was exchanging the solution that they were in, and this is because they're under pressure. And I could nicely see these volume changes. And pressure regulation is quite important for bacteria. They really regulate their pressure. And in other case, this is great, because on an individual cell, I can see volume changes. And if I measure this really, really accurately, I read and I see the network itself is known. The proteins that are involved in regulation of this pressure are known. So I took an analogy. I will be able to figure out exactly how this works, because I can play around genetically with some of these proteins, and the output is the volume, which I can measure very accurately. Similar like this chemotactic network, where you have a couple of proteins and the output is the flagella motor. You play around with them, you figure out the feedback loops, and you know exactly how bacteria is navigating the environment. But it turned out that this other osmoregulatory network, the one that regulates the pressure of the cell, is coupled to pretty much everything else that the cell does. It's coupled to the rest of the physiology. Versus the chemotactic network is quite orthogonal. So when you're studying it, you're quite lucky, because you play around with that network, the rest of the cell is not touched much at all. Versus where you play around with the osmotic network, you mess everything. You have a bit of a, what is a chicken and egg? What is a cause and what is a consequence? So it's a lot more complex network than the one that I thought I was going to be working. The one that we understand, that I thought I was going to be working on something is quite similar, but it was not at all.
Is that where you started thinking, well, there are actually a lot of maybe like physics that we don't yet understand, and we need to scratch a little bit about the surface of that to find, or not really?
No, that is precisely when I started thinking about it, because it became quite obvious that you have one cell, right, that is also an organism. And that cell is regulating pressure, it's regulating swimming, but the way that it's regulating pressure is linked to the way it's regulating swimming. The way it's regulating swimming is linked to the way it generates energy. And all of this, and then it regulates certain intracellular concentration, like intracellular pH, but that too is coupled to the energy generation, because energy generation is electrical as well. So you kind of like realize that there is a lot of variables that are mathematically non-trivially intertwined, but the cell does it. And so the cell somehow maintains all of these homeostasis. So that became a very interesting question. How does it do that? And given how they're coupled, mathematically coupled, these variables, how tight you can have these different homeostasis, maybe one variable, if you keep very, very tight, the others you can't, or something like that. So this became like a really interesting problem in itself. And I sort of, to answer it, I combined the knowledge that I had when I was studying these rotary motors that are involved in energy generation with this pressure regulation that are ultimately coupled. So it all kind of came together to look at what we're looking at now, which is like physiological variables, and especially electrophysiological variables in a life of one cell. It's for once, it's a bit more complicated than in multicellular organisms, because there you have compartments. So you can say, okay, this compartment is going to be in charge for generating energy, and then the other compartment can use that similar type of energy as a signal, not necessarily just as an energy, right? Versus when you have one cell, everything is coupled together. So in a way, it's more complex. So yeah, that's kind of like it all came together to what we're working on now.
So by now you're fully specialized in biological physics, and you mentioned to us that maybe over time, maybe even after Princeton, as you're moving back to Edinburgh, the way people study the proteins in the cells kind of has changed and adapted, and we're now using different techniques to the ones that we used very early on in your research. Could you explain a little bit more about in vivo versus in vitro, and why these are different, and why now people focus on a different version of these techniques to the ones they used in the past?
Right. So this is actually maybe something I should have explained right at the beginning, because the way people use in vivo is different depending on which field you're talking about.
In your field.
So in vitro is quite straightforward, right? You take it out of the cell and you purify a protein, and then you can maneuver it without the cell being, just protein needs to be active. In vivo for us is when a cell is alive. In vivo for many could be in a whole living organism, or in a mouse, or in another animal, right? So the in vivo is the one that gets mixed a little bit. So for us, it means that the proteins are looked at when the bacterial cell is still alive. So I guess before, when this molecular motor field started, most of the proteins would be purified. And then different ways of applying forces would be used, like optical tweezers, which is a sharply focused laser beam that can apply force at the right scale, so that these motors can be maneuvered in vitro. But there is only so much one can do, because you can purify them all, and then you can push and pull, and you get the information that you can from that. But at some point, you now need to start looking at how does this work inside the cell. And then it becomes harder, right? Because the cell is influencing what you're looking as well. So the imaging becomes harder, manipulating becomes harder, but also understanding what it is that is happening becomes harder as well. But people are tackling that as well.
And speaking about cells and their environments, you mentioned that when you were in Edinburgh, you were spinning out a company based on some of this work. Could you tell us a little bit more about that work and that company?
Yeah, so I would say that my lab is very much focused on very basic research. But I learned that by communicating with industry, we can quite effectively translate what we do by answering basic level questions into the application. And the key that I found was, rather than me going to the industry and telling them what I do and expecting that they will understand how is this relevant for them, it worked a lot better when they would tell me what their problems were. And then I would, actually, this thing that we've been looking at, we think that is going to be quite relevant for us. Because some of these osmotic shocks are used in biotechnology, for example, to get proteins out of the cell, right? We can almost burst them up, you open them up, right? So that worked, and that encouraged me to work on the interface as well, and it went from working with industry to companies coming out of the lab. The first one started in February 2020, and it was focused on simply making bioreactors on a scale that was relevant for us in the lab. And it came from a long-standing frustration that to grow bacterial cells to the right stage where you need them to do the experiment can be relatively antisocial, right? They have a certain doubling time. In certain media, that doubling time is different. So you may be waiting for 12 hours. You may be waiting for 8 hours. That is horrible, right? You come in the morning and 8 hours later, you can only start the experiment. And I wanted to buy, both for myself and also for the lab, something that will automatically grow and keep them in the right state. And this was not available unless you had to pay a lot. So this is where the company came in. We're simply making these bioreactors at the volumes that we need in the lab, which 10 to 20 milliliters, which also turned out to be where bigger companies, industrial biotech companies, there was nothing at that scale that was available as well. So it was interesting to them as well. So there's a little bit of luck there, right? We kind of like we were motivated by the scale that we use in the lab, and it turned out that scale was not covered by what was already available on the market, so that the big biotech was also interested in the devices.
And you also mentioned biosensors to us earlier.
Indeed. So there's another company that is right now looking for funding, right in the process of funding. And I would say in the instance of the first one, which is called Augie Biotech, I was certain that we can make the devices to high quality. How many people would buy it outside of my own lab and the colleagues who are interested in it, that I wasn't as certain as, right? And that was a little bit of discovery process, and there was an element of luck in it. But with the other one, I'm way more certain that people will want, because the need to sense analytes in liquid is big. All kinds of analytes, including ions and metals. But the technology is so challenging that I wasn't certain whether it's going to work, right? So what we're essentially doing, we're putting individual bacteria that are engineered to sense an analyte. And this is relatively straightforward to do. Right? And the signal from the bacteria detecting an analyte is sent down to this bacterial flagella motor that is normally there to enable bacteria to swim. And we put a marker on this motor so that the marker is marking the rotation of the motor. And we position that into a customized integrated circuit and micron-sized electrodes. So it's all literally happening at 1 millimeter square. And there is bacteria right next to these electrodes getting electrons out straight from the circuit. Right? And then one can position thousands of these different cells and engineer them to sense different analytes really, really quickly. And the output is always, it's electrical, it's still electrons. So it's very easy to integrate into the current semiconductor-based technology. So yeah, this is the hope is that it will be used to sense many different analytes in the liquid. But technology is really hard. We are positioning one bacterium next to one micron-sized electrode, and it's an integrated circuit that is tiny. But so far, it's really, really going well. So hopefully, it'll keep on going in that direction.
Yeah. Sounds fantastic. And which companies do you think will find value in sensing these analytes in water?
There are a lot of different companies. It was the challenge for the future CEO of the company was really distilling everyone who is interested in it to identify what are the beachhead markets, where to go first. Because a lot of the people a lot of the industry that was interested, and I'll tell you in a second who they are, did not have the necessary technology to implement something like that, something as novel as that, into their current processes. So they need the company to do the packaging around it as well. And when you need to do this, you really need to focus, okay, I can't do it for everyone because they have very different requirements. I need to do it for a few beachhead markets. Some of them is for remote sensing, but in principle, you can send the sensor out, right? You can drop it into the field. You don't need to have it in the lab because low power. But we decided to focus, the beachhead markets are offshore infrastructure. That is the second one. And the first one is really aquaculture, is the fish again.
So after all this time, even though you didn't go to Dresden, you're working with fish.
Yeah, so indeed, there's a lot of labs in the world that maintain fish for research, and they need to be maintained quite well. And also, fish for consumption, like salmon and all of that. And measuring the analytes in water to ensure the quality of fish, both for consumption but also for research purposes, is something that is done in a very manual and painstaking way, right? So they are really looking for improvements there. Offshore infrastructure is an obvious one, right? Because it's quite hard to go out. So something that is in situ and is sending wireless information would be really, really valuable.
Now you just recently moved to Cambridge, and you moved your lab from, you took your lab with you. Why move to a physics department after so much time in biology or bio-related departments?
So I will say that I always view myself still as a physicist while being at the biological sciences department. And I think the reason I moved back is that I was finding that the topics that we are trying to answer are more suited for students who have a physics background, right? And that balance, there will always be about at least 40% of PhD students who were with physics background and postdocs in my lab. But that balance was shifting a bit through the years, and in the recent couple of years, it was going more into someone with a physics background more suited for the kind of questions we are asking. Not all of them, but for majority of them. And I thought that this is going to continue. It's just going to go more in that direction. So that was one of the reasons why I decided to go back. And the one was probably is as a physicist in the biological sciences department in Edinburgh, for example, it was a unique department in the sense that they hired many physicists, mathematicians, engineers within the department, as well as collaborate with the departments, those departments. And some of the vision was, well, biology is becoming very quantitative science. So let's get the expertise from, chemists, physicists, mathematicians, to help us shape that discipline so that it becomes its own heavily data quantitative based discipline. So and I subscribe to that vision, right? But at the same time, the kind of questions we are asking right now, I view as that they hold genuinely new physics, not new physical forces, right? But new concepts that we haven't explained yet. And I think that is more suited for the physics department.
Yeah. And so, what is it about Cambridge and the Cavendish that the like being surrounded by people that are from other field of research and specialties, but within physics, that was the appeal of Cambridge in particular?
Indeed, it was. It was the fact that this is essentially a challenge when you're working on with living matter, because you can't really. Well, you can, but it is still challenging. Living matter throws at you physics from all kinds of directions. You can't decide, I'm going to be an expert in this and this, because you just don't know what is going to hit you, right? There is elements from, soft matter, then there is elements from non-equilibrium thermodynamics, then there is something completely, electrical, where you need to be handling, or like you're building these devices, integrated circuit, so I kind of felt like that this expertise in different areas of physics is precisely what I need to be able to learn fast enough and get the right collaborators to handle all of these problems that life is throwing at you. Because it's quite unpredictable, it's quite challenging, right? You want to be very careful that you don't know nothing about everything, right? So, and you have little control, because what kind of physics comes at you when you're looking at these systems and want to understand how a living cell as a whole works. It's out of your control in a way, right? So, and you don't know the system well enough yet to be able to predict. So I kind of felt that this different physics subdisciplines and experts in it is precisely what could benefit the research.
So now let me ask you, do you have any biochemists in your lab to make sure that you're not missing any?
I'm missing any. Yeah, I'm not missing any behavior. Right now, not. Maybe I need to fix that. It's a good point. Thank you for the suggestion. Yeah.
It sounds like you knew you wanted to be a researcher ever since the moment you chose to study physics. Why was it that this path was so appealing to you back then? Where do you find satisfaction in your role? And also, have you ever looked back and wished you'd done architecture?
Well, let me start with the last one first, because that's for me. The answer is no. I still enjoy, I can read the building plans better than someone who wasn't exposed to it. But that is a quick answer. It's no. Now, about being a researcher and what brings joy, I guess I'm going to come back to something that I mentioned before. It's like an itch. Like certain problems that I wish to solve just bother me. They come out of nowhere, and for some reason I want to find the answer to them, and I can't forget them until I solve them. And that is really what for me, it drives research, right? It's literally, you feel like you need to scratch, and until you scratch it, you can't let go.
So it's that feeling that decides the question that you
it's
choose.
Precisely, it's like, it's not rational. And you rationally go about solving the problem. But what I find that how I choose the problems is not a rational problem. It's just something that, okay, this is something that not only I'm excited about it, I'm excited about many things that I don't necessarily wish to solve myself. I'm excited about it, and I do wanna, it bothers me. I want to solve this.
And do you have any questions that you really want to solve that you haven't yet, that you've left open?
Not only the ones that are left open, the ones that I haven't had the opportunity to touch. It's just like it seems to be an enormous source of problems that I do want to solve. And the ones that I haven't solved, they keep bothering me, and I come back and I hope that I'm going to solve them. And although there are probably going to be many open questions by the time I'm done with it as well. So yeah.
Do you keep track, or are they all?
Oh, I do keep. They keep track by themselves. They're kind of like lingering somewhere in the back of the mind, bothering me that they haven't still been solved. So yeah.
We're going to end there, but you said before that in science, nobody really knows all the answers, and that this is okay. You have to live with the fact that the best that you're going to do is an educated guess. Would that be the advice that you would give? Like, what would you say to young, inspiring scientists, and in particular experimentalists that are thinking about going into this kind of career?
I would give this as an advice just to set the expectations right, because if you're aware of it, I find that you're handling it, you're not surprised by it. It is the very nature of it, right? But at the same time, what you find is the answer, and the people that you're going to be interacting in order to answer that just takes you to places you would never go otherwise. And you can never predict where it's going to be. Like, it's that unpredictable. And this is really exciting, right? This is really, I would say this element, like, it's tedious sometimes, right? It's many times you get stuck. And enjoying it, that's part of the process, right? If it was easy, it would probably been answered before. Right? So getting stuck and getting, and trying to disentangle yourself, that is something that is part of the process as well, and it's often there. So I would also I would mention this, so just that everyone is aware that it's sometimes quite slow, right? For a reason. But I guess that getting stuck, I find motivational as well, right? So kind of like, okay, this way, that way, try this, try that, like, and try to get yourself unstuck, right? That is like, if that is something that motivates a person, that is probably a right career choice.
So it takes a bit of imagination.
I guess imagination, but also perseverance. I would say, yeah. Both.
And patience.
Patience is quite important. It's not necessarily a fast process. Sometimes it is, but most of the time it's not a fast process. It's definitely a marathon.
And a bit of resilience as well.
Yeah, indeed. There are plenty of awards, but their resilience is important.
Excellent. Thank you so much for being here today and for your time. That was lovely.
Thank you for having me both.
Thank you to Pilizota for joining us today. As always, if you would like to learn more about what we discussed in this episode, and more generally about our work at Cavendish Laboratory, please have a look at the show notes or go to our website. If you have any questions you would like to ask our physicists, head to social media and tag us with the hashtag #PeopleDoingPhysics. This episode was recorded and edited by Chris Brock. Thank you for listening to People Doing Physics. We'll be back next month.